And what I'm actually going to focus on in the 3 talks I'm giving are really sort of new ways we can think about translocation. And one of those is about assisting gene flow, and the other one is about assisting colonisation. And in our case, in the southwest of West Australia, which is where we have one of the most sort of profound changes in the climate in Australia with extensive drying since the 1970s, both these, those species that occur in this bioregion are really going to have to adapt very quickly to a different hydrological regime, and there may be ways that we can assist that adaptation. So the first talk is this one, 4.6.1, about assisted gene flow to increase resilience of frogs to a drying climate. And there's some context for this work. Back in the 1980s, you know, that was around the time that Australia was starting to think about climate change. We were calling it the greenhouse effect. And CSIRO actually commissioned a really interesting book called Greenhouse, Planning for Climate Change. And it asked some of the most sort of active and influential ecologists of the day to, I guess, make some sort of predictions about what might be the impacts of climate change on our fauna and what we might potentially need to think about doing. And so G.W. Arnold, a West Australian ecologist, wrote about possible effects of climate change in Western Australia. And one point he made is that the redistribution of species, especially those that are less mobile, are going to be hindered in the more agriculturally developed parts of Australia because of their isolation in remnants of natural ecosystems. And of course, he says this very much in the view of being a West Australian, where about 90% of the West Australian southwest has been cleared for agriculture in the last century and even up until a few decades ago. So a lot of fauna is now effectively isolated in remnants. And actually, he went on to actually be specific about some of the things he was expecting to see in terms of which species are going to benefit from climate change and which ones might not. And the species that he actually all predicted to be negatively affected were things like our terrestrial breeding frogs. And that's the species system, I guess, that we're going to be focusing on particular in this morning's presentations. So Australia has a very rich frog fauna of about 250 species, and around 20% of those, or maybe a bit higher, don't lose water for breeding. They tend to lay their eggs in terrestrial locations, and obviously as our climate's drying, those that nesting success might become increasingly less successful if those soils are too dry for their survival. And that's partly what we're going to focus on. So I've been working on this species for a couple of decades now. It's called the crawling froglet, or crawling frog, Pseudophony guntheri. And one thing I noticed when I was doing research in the early 2000s, I was working in some pretty terrible years of low rainfall. So this is just showing you some of the climate anomalies in the early 2000s relative to the historical records in Wandoo woodland, which is a beautiful type of eucalypt forest where this species occurs in these seasonal drainages. And I was noticing that in the years of low rain, the frogs were not breeding. The males were struggling to have, I guess, much of a calling season, and maybe those that did breed were having very low reproductive success. And what happens in dry years is that frogs, they may not be able to be sort of well enough hydrated to make their calls to the females. And also, if they do manage to mate, then the eggs will become very small and shrunken. They basically suck up the water from the dry soils, and they basically, the embryo that develops inside has very little room for movement and often can hatch in quite a poor state, as you can see here from this sort of illustration on the right, which shows the impact of different water tensions on frog development. So I was quite concerned that, I guess, dry years and successive dry years might start to threaten these types of species. And so that's what gave us, I guess, cause to think about whether assisted gene flow could be something worth exploring in this species. So the crawling frog is not a threatened species, and it occurs broadly across the southwest of West Australia in these little fragmented populations. These are just records from the Atlas of Living Australia. But if you look at its distribution more broadly, you can see it expands a wide range of rainfall isoheights and a broad range of temperatures. So obviously across its range, there's going to be populations that have been evolved for a long time and adapted to different types of climates, perhaps the drier, warmer climates that we're going to see transitioning down through the southwest. We know that this population is losing some genetic diversity. We've got some examples of that from genetic studies we've done. So that means, I guess, that their potential to adapt without assistance is perhaps somewhat constrained. So we wanted to explore this idea of assisted gene flow, which is managed movement of individual animals, or perhaps just their gametes, between populations within the species range to mitigate maladaptation. And so to do that, we first wanted to understand, well, how much variation is there in this species in terms of its tolerance to desiccation? So Tabitha went and collected a whole lot of frogs from these different locations across a couple of rainfall gradients, north and south. And then we looked at their ability to rehydrate and dehydrate depending on the soil moisture, and basically showed that, as we predicted, the frogs that came from the lower rainfall areas didn't lose water as readily as those that came from more mesic areas, and they were also better able to rehydrate. We then went on and made tadpoles. We basically created crosses of the adults that we brought in. We bred them in the lab, reared up their young, and then we reared up the eggs on 3 different soil treatments, simulating the challenging sort of conditions of a very dry soil type, which is these white dots here. And the main point is that this population 5, which you can see is consistently circled through this figure, they did really well on dry soils. So it's pretty clear out there that there is variation in traits related to desiccation tolerance. And what we wanted to explore is whether or not, if we introduce those genes into perhaps a population that is more evolved under wetter climates, would those genes offer some, I guess, greater resilience to their offspring? So that's essentially what we did with this project. We went out and collected frogs from very low rainfall environments, which look something like this. This is up near Yalgoo in the north, in the sort of Gascoyne region of Western Australia. So we collected adults of males and females, brought them back to the lab, and we also collected frogs from high rainfall environments. And so that was really quite an amazing thing to be able to do. Tabitha did this in the year 2017. Luckily, in that year, there was quite synchronous rainfalls across the range of this species, which meant they all bred more or less at similar times. So we could actually try and use that opportunity to see if we could cross populations. So what we did is we went to the first population, population A, which was the wet population, collected about 10 or so males and 10 or so females. And then in the laboratory, you can basically create crosses using mixing up the gametes in strategic ways to make lots and lots of family groups that have this particular genetic makeup, A females and A males. Then we did exactly the same in a drier population inland, population B. We did the same in a population up north of Geraldton, population C. And then in population D, another dry population, we didn't get females at that site, but we got males. And then the other thing that Tabitha did is that she then started to cross all these populations together. So population D, the sperm from those males we used to fertilise eggs from populations, the wet population here, A, C and B. Population C males were fertilising eggs from A and B as well. Population B males were fertilising eggs from C and A, etc. So we basically created a whole range of crosses, which are called reciprocal crosses, because we did the crosses in both directions when we had both males and females. And so that allowed us to create lots and lots of different family groups with different genetic makeup. And I think there are about 3,000 individual eggs and embryos in this experiment. So what we did then is those eggs were created and then we put them onto wet or dry soils. So wet soils being kind of benign conditions, dry soils being something quite challenging. And then we reared them up for a couple of months. That's what happens in the wild. They incubate on the land for a couple of months, and then after winter rainfalls, these nest sites flood, and that's when hatching occurs. So we simulated that by simply flooding the eggs. And then we looked at how did those tadpoles vary, and did they have traits, I guess, that were different and perhaps indicating that they were more tolerant of those dry conditions. The other thing that was really great about this study is we also knew about the genetic similarity of the populations that we crossed. So this figure here shows where those locations were. It shows the geographic distance between the sites that we crossed, and these are very big distances, as you'll see, more than 500 kilometres. And these are genetic distances. So the darker the colour, the more dissimilar the populations are. And here's an nearest neighbour tree showing some genetic structure based on SNP data. You can see populations C and D up here are quite closely related, as are A and B, but there's quite a distinct difference between this branch here and this branch here of this species. So our expectation, I guess, is that these are quite big genetic differences, and we might not see a good impact of crossing. We might actually see something like outbreeding depression, where you get a maladapted offspring. And so there were a lot of results to analyse, which I'm not going to show in this presentation, but just to summarise what happened, this is sort of a schematic of the crosses that we did. So within the crosses, these are the grey results. Basically, you get the expected phenotypes. As we'd already tested in the previous experiments, we knew that these guys were less tolerant of dry soils than these ones up here. And then we looked at the crossing. And what we did is when we crossed the north and south populations in particular, every time we did that, we got probably outbreeding depression. We didn't get high hatching rates, or we had high rates of malformation. So that suggests that crossing at that scale isn't going to be effective. And one of the reasons we think it didn't work so well is because we've actually now got evidence that there's probably differences in reproductive strategies across the north and south of this population, this species, potentially related to the reliability of rainfall in those areas. So I think that's mean there's been quite strong divergence on things like sperm characteristics. But what we did get is this really fantastic result where we mixed populations B and A, and we actually got fitter offspring, so that they were actually performed the best of every group in this experiment. So what happened is that the tadpoles that were produced by sperm from population B, which was a drier population, with the eggs from population A, which was our wettest population, those offspring performed the best of all. And that's a really fascinating result because we did not get it in the other direction when we crossed eggs from B with sperm from A. So it suggests that perhaps the direction of the cross is important. It probably hints at something like interactions between mitochondrial and nuclear genes that gives that sort of adaptive response. So we did get a really good outcome in one case. And I think that gives us, perhaps it was an unexpected result in the sense that we didn't see outbreeding depression across the board. We did get the sort of targeted gene flow that we were hoping to get. And it worked when those populations were at similar latitudes and perhaps a bit more closely related. And so now I guess the next steps of this project is we don't have genomes for Australian frogs in any great extent. There's actually frogs as a group don't have a lot of annotated genomes available, but that's changing quite rapidly. And for example, the corroboree frog, which is in the same genus as this species we worked on, has a genome under development. And so in time, we might actually be able to target traits quite precisely that we're interested in introducing to some populations, and to see if we can get this as an actual practical method that could be used in the field. So I think the conclusion of this work is that assisted gene flow could work for frogs. The real value of doing this work for frogs is the fact that they're external fertilizing, so you can actually manipulate breeding lines very easily because you can access the gametes and create individuals outside of the animals. And you can also do it at quite a large scale. But of course, it's not going to be something that we'd be ready to apply in wild populations right now. You'd need a fair bit of time to actually go and map that variation that you're interested in selecting within your population. And you'd also, I think, really want to test those crossing outcomes in the laboratory first before going ahead with trying it in the field.
Yeah, so hi everybody. My name's Emily Hoffman. I'm a PhD student at the University of Western Australia, and just actually recently handed in my thesis last week. So you've got all of the findings here today in a, I think, 7 to 10 minute presentation. So I'm not sure how I'm going to go with squeezing 4 years in there, but I'll do my best to be concise. And yeah, really happy to chat after about anything that I've missed, or flick on the papers that we've published from this work. Yeah, so first of all, I just want to say that this project, my PhD was really centered around this project 4143. Improving the conservation outcomes for the critically endangered white-bellied frog. But it wasn't just me. I was working with Nicki, who just spoke, and Matt Hipsey at the University of Western Australia. But it was a really collaborative project. So we're working with Western Australia's Department of Biodiversity Conservation and Attractions, their Parks and Wildlife Service. A big shout out to Kim Williams, who I think is here today, and Chris Taylor, and also the Perth Zoo team at the Native Species Breeding Program. They had a large role in this project, and we also received support for my scholarship from the Forest Research Foundation. So white-bellied frogs are those guys on the left there, Geocrinia alba. They're pretty small little frogs, only about a couple of centimetres long, and they're by far Western Australia's most threatened frog species, so they're listed as critically endangered. And their sister taxa are actually vulnerable as well. So on the right there, we've got the orange-bellied frog, which is Geocrinia vitellina, and they're listed as vulnerable. And these two species have really highly restricted ranges. So they're both endemic to the southwest, but you can see those tiny distributions are pointed with little arrows there. They're only about 10 kilometres across in radius, so really restricted. And they also coincide with that region of the southwest that's experiencing significant warming and drying. These species have really specialised habitat requirements. So they're terrestrial breeding frogs, like some of those ones that Nicki was just describing. So they actually don't require water to breed, but they breed in the sort of moist, boggy soils that surround seepage areas and ephemeral drainage lines. And they occur in those unique habitats. So they have quite a patchy distribution within their restricted distribution. And they don't seem to move from those patches. They've got really low rates of dispersal. And we were particularly interested, I guess, in white-bellied frogs because they're critically endangered. And that's because half of the known populations have become extinct in just the recent decades, in the last few decades since they were discovered in the 1980s. And populations are continuing to decline. And the Department of Biodiversity, Conservation and Attractions and PERSU have done an amazing job actually doing some fantastic translocations. Some have been incredibly successful, but to some other sites less so, and it was unclear as to why perhaps there was such mixed success. So my project, or our project, was really about how can we guide conservation actions for range restricted frog species that are restricted to a drying climate. And I guess the overall objective was to try and better inform translocations, to try and increase their success, and also to consider potentially assisted colonisation, like Nicki was just describing. But we sort of stepped back a little bit and wanted to fill some key knowledge gaps before I think any of those decisions could be made. And they included really these three main questions that I've sort of summarised here. And that's why are white-bellied frogs declining in some areas, trying to understand what the sort of causal factor is there, what environmental factors are associated with population persistence, so can we try and understand more about their habitat that they require, and just how might these frogs be impacted in a drying climate. And we did this by using a number of different methods. I'm not going to go into detail into what they are, but I just wanted to give you an overview, I guess, of just how we approach these questions. And so we looked at their microhabitat requirements, so really fine scale environmental features and if they're associated with persistence or extinction of sites. We looked at their ecophysiology, so actually taking frogs into the lab and trying to determine their hydric and thermal thresholds, and then comparing that with their microclimates in the environment and just how close they're living to their current physiological maxes. And we also looked at their breeding phenology, so whether or not breeding cues, if the cues for breeding were actually linked with certain environmental conditions, and that they might be ones that are changing with changing climate and drying climate, and how much variation there was within their really small range, was it all the same or not? And lastly, trying to identify, I guess, ecological refuges, so areas where these species might be able to persist through time, and looking at landscape scale requirements to do that. So first of all, looking at why white-bellied frogs are declining in some areas, I'm just going to really just hit home with the major findings. I don't have a lot of time, so I'll just quickly summarise each of these questions. So the first one, we think that drying and warming conditions are the principal driver of recent white-bellied frog declines. So we found when we compared sites of where frogs were and where they used to be, that frogs have actually disappeared from sites that are now significantly drier during those drier months, so the summer, autumn months. We also found when we looked at the physiology data that frogs are experiencing physiologically stressful conditions in those summer months in really extreme hot and dry years, and that's under current scenarios. And looking at the breeding, we also found that breeding periods in those sites that have drier conditions, they're actually shorter, and that means that there's potentially reduced breeding opportunities at those sites. When we looked at the environmental factors that were associated with persistence, we identified a number of things that were on different scales. So for example, on a microhabitat scale, we found that the site hydrology, so that soil moisture and temperature, was really important. Certain ground cover and soil characteristics. And that also was important for not just natural populations, but translocated populations as well. We could sort of retrospectively predict whether or not a translocation should be successful or not based on, for example, the hydrology. And that was quite, we found that to be quite important. And looking at a landscape scale, we found things like the amount of vegetation cover at a site and the fire frequency was important for site persistence, as well as site topography for those orange-bellied frogs. And like I mentioned before, we also defined their physiological thresholds. And I guess I didn't really want to get bogged down in the detail here, but I just want to say that we have a suite now of knowledge now around, I guess, what the optimal environmental conditions are and their physiological needs, so that we can then help to inform things like reintroductions and maybe evaluate whether there are potential areas that might be optimal for assisted colonisation if similar systems exist elsewhere. And likewise, identifying refuges, those areas where the species might be able to persist long term. And lastly, how might these frogs be impacted then by a drying climate? Well, I think we've definitely validated that climate change is a really immediate threat for the white-bellied frog. We know that they have low tolerances to hotter and drier conditions. The microclimate conditions that we're looking at now are already breaching the physiological thresholds of these frogs in some areas. And it seems that when you look at the landscape, the microclimate conditions that they require are quite niche and they're really rare in the landscape. And it seems like they're shrinking. And also looking at their breeding phenology, the fact that their length of the calling season was so strongly linked with soil water potential at sites and temperature, I think all of those things combined, it sort of has quite significant implications given that the drying and warming conditions of the southwest are predicted to continue. And actually speaking with some hydrologists and groundwater specialists, they highlighted, I guess, that these sites, because they're sort of at that interface between the terrestrial zone and the aquatic zone, they're quite susceptible to these changes. And for example, a 1% decline in rainfall actually equates to a 3% decline in runoff and groundwater recharge. So it's slightly exacerbating the drying conditions actually at these sites. So I came up with a few, I guess, summary points, or what I thought would be the future research directions following on from this work. And I think one of the key ones is to monitor some key refuges. So we actually identified some sites doing this work that these areas seem to be quite stable through time. So they maintained a really appropriate soil moisture regime during the year and appropriate temperatures. So I think those sites could be really important for the species persisting into the future. Likewise, we can use all of this new information to be strategic in the selection of translocation sites. And I know that the staff at DBCA are already doing this and doing a really great job with that. Thirdly, perhaps even considering some sort of novel engineering actions to address the threat of drying conditions at some sites. So for example, some people have suggested strategies such as environmental watering or forest thinning that can help to, I guess, mitigate the drying at some sites. And lastly, we've got an incredible amount of data now on the microclimate conditions at these sites, which are quite different from the terrestrial conditions. And so using that and our knowledge now about the frog requirements to predict microclimates for these frogs under future climate scenarios. So this project was really looking at current scenarios, but I think it would be really interesting and a clear next step to go for this species. And lastly, just to finish, I think whilst it was a really species-specific project, as a lot of threatened species work, is necessary to be, I think there are some broader implications for some other amphibians, given the changing climate. Most broadly, I guess this project's really provided some empirical evidence of how amphibians might be affected by a drying, warming climate. And this could be immediately relevant for some of those other species that Nicki was talking about earlier, actually, as terrestrial breeding frog species that are also threatened by a drying climate. So these are species that have narrow climatic niches, so they're constrained potentially to small ranges on mountaintops, or reliant on similar sort of seepage systems and ephemeral temporary streams and drainage lines. I think something that's also in the forefront of my mind after the Black Summer fires in Eastern Australia is just what I'm trying to understand more about this relationship between drying microclimates and fire. And I think it's really important that we identify areas that might be more resilient to drying into the future. And lastly, but certainly not least, I think the methods that we've developed in this project could have immediate application for Western Australia's only other threatened frog species. So there's 3 in Western Australia, and this is the sunset frog, which has recently undergone substantial population declines, but a lot less is known about them. So we're not quite sure about their detailed habitat requirements on a fine scale, what cues they have for breeding, and just how vulnerable they might be to climate change. But we certainly think that they're highly threatened by fire and drying climate. And yes, a big shout out just to all of the many people who've contributed to this project. It's been really collaborative and I think a really successful one. So thank you.
Okay, so I suppose I'll just first of all acknowledge the traditional owners of the land on which I stand, which is the Whadjuk Noongar people, and then I'll crack straight in. So this talk doesn't involve any cold-blooded species at all, so I'm sorry about that. But it was really focused, quite species specific, and focused on understanding some genomic variation in the Western ringtail possum, working quite strongly with Sarah Comer and Adrian Wayne from DVCA. So a little bit of background behind the ringtail possum, or noyer, as it's known in the traditional tongue. Smallish nocturnal arboreal folivore. It's endemic to southwest Australia, and since European colonization, there's been around about a 90% reduction in range. Usual suspects there, foxes, cats, and dehabitation and habitat reduction has sort of led to a lot of this. So as a result, and also due to predicted continual declines, it has been uplisted to critically endangered by the IUCN and BC Act in 2018. A little bit more background. So as a result of some of this, the species has been separated into 3 management zones. These are the Swan Coastal Plain Management Zone, the Southern Forest Management Zone, and the South Coast Management Zone. This is sort of a distribution map here to show you what the landscape looks like currently, and also shows some translocation sites as well. So the estimate is that there was in the hundreds of thousands of possums before Europeans arrived. That has reduced quite significantly, and the most recent estimates now put that number around about 20,000. There's been 8 approved translocation sites for noyer. Most of these, however, have been unsuccessful, and only 3 of those still remain. Of note, around about the late 1990s, there's been some massive population reductions across the board, really, but specifically in this Upper Warren region, where we've seen up to 95 to 90% reduction in numbers, which is quite dramatic. And further declines are predicted for the species. And a paper I refer to there by Malloy et al. Did some distribution modeling and some climate modeling as well, and predicted that as a result of climate change, the habitat impacts on habitat, it's pretty dire news for the possums who are so reliant for their nutrition and water from the leaves. So. But not only all of that, there still remains, which I found quite surprising as a conservation geneticist, quite a strong paucity of genetic data on the species. And that's quite a problem, because what we're really looking at here is a situation where, as these populations are crashing, there's a good chance we could be losing some genetic diversity irreversibly before we've even documented it. So here's just a couple of figures to show you some of that empirical data. These are produced by Adrian in 2012, and what he was doing was using some spotlight sampling, and sort of you can see here quite clearly just how the numbers that he was finding were really crashing around the late 1990s and early 2000s. So this plot here is from one region in the Upper Warren, and around the Kingston Park area. He also looked at another region in the Perup, and again you see the same sort of trends with this massive crash in numbers in the early 2000s. Okay, so what sort of questions then were we interested in this project? So our questions were really, what does a noyer genetic diversity across the remnant range look like, and also how have these population crashes affected that genetic diversity? Is there any evidence for some distinct genetic lineages or units within the species? And if there are, what are the relationships between these groups? And can we identify any association with environmental variables? As you could see, the range sort of spreads from west coast of WA down to east of Albany. So across that region, there's quite a difference in vegetation as well as rainfall and temperature. So it'd be interesting to associate some of those variables with that genetic variation. And also, we're interested in what would the population trajectories be, and try and understand a little more likely threatening processes for the populations. So how do we approach this? Well, from the map, you can see there's quite a range of quite a long area to sort of cover. So instead of going out and doing targeted sampling, I decided to do passive sampling. So I was really trying to access samples from historical collections, and any gaps would then be filled with opportunistic sampling, which is often roadkill and required a lot of help from the community. We sent the tissues off for SNP genotyping, which is a type of molecular marker, which is quite powerful for this kind of work. And we used DART-seq for that. We investigated some bioclimatic variables across the range and looked for some trends, and then we used population viability analyses to try and understand some threatening processes, and we focused that really around Upper Warren. Okay, how did we do then? Well, after it all was said and done, I managed to get 515 samples from 57 locations, and they're sort of spread across the management zones, as you can see here. Each sort of little red dot on this map is sort of showing where we got samples from, so we've got pretty good coverage overall from the remnant range. Of those 515 samples, we genotyped 305 from 52 locations, and this gave us around 23,000 SNPs to work with. So you can sort of see the sort of power that you can get from this sort of analyses. So I'm going to crack into the results for the rest of the talk now, really. This first graph is just sort of showing you genetic diversity that we found using some of those markers. And in this graph, the green colored bars represent the populations from southern forest. Red represents Swan Coastal Plain, and blue represents the south coast. And if you sort of just looked at it from a color perspective, you'd sort of see that most of the greens are headed towards the right of the graph, and that's really indicating that there is higher genetic diversity within the southern forest localities. And if we go on and look at another type of genetic diversity, in this case, I'm looking at private alleles. So in this particular case, again, the colors are the same. Green is for southern forest, blue is for south coast, and red is for Swan Coastal Plain. You can kind of see that towards the right of the graph, we see the sort of step increase in private alleles for 3 localities. And private alleles really indicate the amount of genetic variation that is unique or specific to a particular area or region. And these 3 localities on the right are Malloy Island, Pemberton, and Two People's Bay, and all 3 of these have actually not been included in any previous work or assessment of genetic variation within the species. So this would sort of indicate that these 3 localities have quite a lot of genetic variation which is unique to these localities, and that would indicate it's quite important conservation value. Okay, this is quite interesting because a lot of the work that has been done on conservation efforts for the Western ringtail possum have kind of focused historically on that Swan Coastal Plain region and less in those other regions historically. So it's interesting that we're finding that some of the more diverse populations are actually in the other regions altogether. Now, what do we do with some of this variation? Well, I wanted to try and define some genetic clusters and try and understand what sort of level of structure was going on within the species. And we used several techniques, and this is just a summary of a couple of the techniques. This is a structure plot on the right, if anybody's familiar with that. This tree on the left is actually a genetic distance tree. It's a phonetic tree. And using a few different approaches, we were able to sort of show that there are at least 8 genetic clusters in the Western ringtail possum. And you can see that there are 2 in the management zone of South Coast, there are 2 in the management zone of Southern Forest, and around 4 in the management zone of Swan Coastal Plain. So yeah, overall, we're sort of showing that there are at least 8 genetic clusters. There may be a 9th, which would include this one locality of Pemberton. So this is more of a summary of those clusters. I'm not going to go into them in any detail because we're not all possumologists. But what I am interested to sort of describe is this question of how clustery are the clusters. I'm not sure if that's really a technical term, but what I'm really trying to get at here is from an evolutionary biology perspective, although we do have these 8 genetic clusters, how important is that? So what I'm really talking about is trying to find out, do we have any distinct genetic lineages between which there is no gene flow, which might indicate that those genetic lineages or units are on separate evolutionary trajectories. And to do that, I use something called a fixed difference analysis. And whenever I used slightly less than the most stringent criteria in this analysis, I was able to show that there are possibly 3 operational taxonomic units within the species. And these are defined by 2 Peoples Bay region, which is just east of Albany, the lower Blackwood River catchment, which is in the southeast of the Capes region, and then everything else, I could not show that there was any barrier to gene flow between those other sub-regions that we've described. So possibly here, 3 operational taxonomic units. We were also interested in trying to look at some environmental variables across the landscape. And for this, I worked with Stan Mastrantonis, and we looked at various variables, and I've just shown a summary of 2 here. We've got annual rainfall on the left and mean annual temperature on the right, and you can see that there's some quite interesting gradients across the landscape. In particular, in the annual rainfall figure, you can see that there seems to be this line of demarcation that kind of separates very low rainfall from higher levels of rainfall, and that line seems to run right through the southern forest management zone. In particular, this region corresponds to the Upper Warren population. So there's maybe an interesting finding there if we remember that it's the Upper Warren population where we have evidence for very strong population crashes in the late 1990s, around 95 to 99%. Okay, so what we ended up doing with some of these environmental variables was trying to find out which would be the most important for predicting genetic cluster inclusion. And we ran something called a random forest analysis, and we were able to show that various combinations of environmental variables were quite good at predicting genetic cluster inclusion. So each color on the map on the right indicates one of those 8 genetic clusters, and within those 8 genetic clusters, there are certain combinations of environmental variables which are quite good at predicting whether or not you'll belong to one of those genetic clusters. So it's an association of environmental variables with the genetic clusters. And the 3 most important of those environmental variables that we were able to show was mean summer rainfall, mean summer temperature, and the evapotranspiration rate in February. So these were quite important factors in those cluster predictions. Okay, so moving on to the last couple of results slides, we also want to look at some population viability analyses, and for the reason of trying to understand some of the threatening processes and maybe help understand a little bit about why some of the translocations have been unsuccessful in the past. And this was quite an interesting finding. Although we had not great certainty on some of the life history parameters for the species, I was still interested in finding out the potential of these species to crash or to persist, dependent on various variables that we wanted to test. So for example, here I'm showing what the effect of adult mortality may have on the survival probability of the Upper Warren population. And what I've got here, I'm testing various levels of annual adult mortality, and showing that over time, what the impact that has on the probability of survival of that Upper Warren population. And if you can kind of see, it's quite dramatic, using the only empirical estimate of mortality for the species for this region, and quite across the whole species, which was actually calculated by Adrian Wang, we get values of mortality between 40 and 50%. Now, if you look on this graph, you can kind of see that that would be around this region here, and that would predict that at those levels of adult mortality, you would expect these populations to go extinct within 50 years. Now, it's believed that that level of adult mortality was actually from a particularly bad year, and there's quite a lot of invasive predation going on at the time. So if you remove the invasive predation from the equation, and we came up with an adult mortality of around 23%, at that level of annual adult mortality, we would come over here and we can kind of see that there's a survival probability of about 30% over 100 years. So it's not looking great for this population overall. We also want to look at catastrophe frequencies, so the frequency of stochastic environmental events and what impact that might have on probability of survival. And here it's quite a simple graph, just looking at 3 different frequencies of, say, something like a drought. And here we modeled it to be quite modest, have quite a modest impact on survival. And we looked at frequencies of 0%, 10%, and 20%. But again, this is just slight increases in mortality and slight decreases in reproductive output. And as a result of that, you can see these quite strong crashes in survival probability over time. So some quite dramatic results coming from that. Okay, so then just to summarize then the work, so what we can sort of say from this work is that we've found novel genetic variation which will likely be of high conservation value and give the species a lot of standing variation and help it adapt to any changing climate going forward. We reckon that there are now at least 8 genetically defined subregions within the species and possibly a 9th. And we also are suggesting that we rename the Swan Coastal Plain Management Zone to West Coast Management Zone, basically because the Swan Coastal Plain is only one of the subregions of that management zone. We have high genetic diversity in Upper Warren. This is quite key because this population is continuing to decline, and there's a good chance that we're losing some of the key genetic variation for the species if this population continues to decline. We've got evidence for 3 operational taxonomic units, and we're finding some association with environmental variables. So the PVA work is kind of indicating that Upper Warren population is particularly sensitive to increases in mortality and decreases in reproduction. I can talk about more about that later. So implications of this work would be that we now have a comprehensive coverage of genetic variation across the critically endangered now year. This will help prioritize populations for management in terms of the ones which have particularly low variation, the ones that are showing inbreeding, for example. We've got variation within and distance between the populations, as well as the knowledge of the bioclimatic variables will be useful for ongoing management and optimizing translocations. However, we do urgently need quantification of the impact of stochastic environmental events on population dynamics, and overall, some more of those life history parameters, we need to be a bit more certain of them. We need some greater sample sizes for some of the more remote localities, and ongoing monitoring is essential, including genetic monitoring to see if we can find any evidence of inbreeding depressions in some of the populations that have lower genetic diversity, and also a consistent monitoring across the species so that estimates of census sizes, for example, can be compared and are robust. And with that, I will finish, and thank you very much. And thank you very much to all of these people as well, without whom this work would not have been possible.
Okay,
that's good.
So thanks everyone. So this is you're going to get me to finish off, and I'm changing direction now to talking about reptiles, and we're going back to the theme of assisted colonization. So this is a project that's been going for a long time, for more than a decade now, where we've been really thinking about how to assist this species to persist in wild environments as our climate is getting drier, in particular in the southwest. So the swamp turtle is an iconic species in Australia, partly because its legendary rediscovery in the 1950s. It was thought to be extinct, but was recovered in the 50s and has been really a flagship for species conservation, really good management and attempts at recovering. But we still only have around about 70 or so individuals in the wild, and we have a lot of swamp turtles now also being bred in captivity for translocations. These are shots from Gerald Cookling at DBCA, just showing you, though, I think, a great illustration of just how vulnerable this species is and how much habitat it's lost. So the only two reserves that the species occurs naturally is Ellenbrook Nature Reserve, which is a little bit larger now, right on the edge of the Great Northern Highway, surrounded, as you can see, by sort of light agriculture and industry, and then Twin Swamps Nature Reserve over here. So very small patches of habitat. And of course, with climate change, this species has very little chance of changing its distribution just because of fragmentation of its habitat. There would have been a lot more habitat for this species, but by the time of its rediscovery, it was already too late. So this is like just a projection showing you, I guess, what's expected in terms of climate scenarios for Australia. It's kind of a teaser report put out by the IPCC, basically pointing out that the trend that we've seen of drying since the 70s is very likely to continue into the future. So the western swamp turtle is a species that lives for about 100 years, well, we don't know how long, but at least 80 years and potentially as long as a sort of sea turtle lifespan of 100 years. And it's got basically very low reproduction rates, which doesn't really necessarily matter when you live for a long time. But we're concerned that with climate change, that the reproduction is going to get very difficult for this species. Now, the swamp turtle lives in seasonal swamps, and in most of the year, the swamps don't hold water, and that's when animals go into a form of torpor called estivation, where they save energy. But as soon as the rains break in the late spring and into winter, then the swamps start to fill, and then the turtles then are active, go into the water and spend a lot of time foraging for tadpoles and invertebrates, and that's when mating occurs. And that's when females are accumulating energy for reproduction. But what we've seen in the swamp turtle is that in good years, when it was first discovered in the 50s and 60s, hydro periods could be about 7 months long near Perth. Now, in more recent decades, we've seen shorter hydro periods of maybe 3 or 4 months. And you can do something to extend hydro periods by things like pumping water, and that's indeed what managers have done to try and extend the time that the turtles have got to grow each year. But we're increasingly concerned that with less and less rainfall and declining groundwater, that those sites are going to be very difficult for species to reliably reproduce, to recover those populations to sort of better sizes. So that's really the motivation for why we started to think about assisted colonisation for the western swamp turtle. So this species has had a recovery team in place for a long time and written up to its 5th recovery plan. And the recovery plan back in, I think it was the 4th one back in 2010, clearly argued that we need to be thinking about climate-proof translocation sites for the western swamp turtle. This species has already had successful translocations to the north of its current range, but those areas are going to be, if anything, slightly drier than the current range because of, obviously, rainfall gradients are increasingly dry the further north you go. So we got involved in this research around 2008, I think, and we started to work on ways that we might be able to predict good areas for this species into the future. The difficulty for doing this with the western swamp turtle is because it only has one essentially known distribution point, you can't use a statistical model to predict its future climate space. So in the end, what we've done is we worked with hydrologists and biophysical modelers to come up with a kind of mechanistic way to predict where its habitats might be in the future. And we ran models with different simulations of future climates, which will be drier climates and slightly warmer temperatures. And to cut to the chase, what we found is that areas right down here on the south coast of Western Australia, in about sort of around 2030 and beyond, should be really quite ideal habitats for the western swamp turtle. So with this in mind, we've presented a lot of these results to the recovery team, and we got their endorsement and then the endorsement from the West Australian DBCA to go ahead with some trials of assisted translocation and assisted colonisation. And remember defining that earlier for the frog talk, that means that we've deliberately introduced them into areas where we have no evidence of their historical occurrence. And we were sending them about 300 kilometres further south. So we began the trials in 2016. And we got some interesting media coverage, including coverage in Science, mentioning that we're setting a controversial precedent by doing this introduction. We weren't relocating the tortoise, I should need to emphasise that. We were doing an introduction from captive breeding. But they were basically making the point that this is a flagship project because it's actually taking action and addressing climate change specifically in the choice of translocation site. And a lot of people who commented on this trial, perhaps this is an example of a quote from this story, saying people are going to look at this trial and they're going to say, what about my species? I wouldn't want to spin that roulette wheel many times, basically pointing out to the discomfort that people still have about novel ecosystems and introductions into novel areas. So with that in mind, we always went in with a sense that this is a trial. And so we've been doing actually 3 trials now, trying to learn from each one and to sort of refine what we're learning, to ask new questions. But we're hoping to get to the point where we actually might sort of go ahead with an introduction into these southern areas. So in 2016, we had a trial which essentially covered these 3 regions. We released animals into an existing northern translocation site up at Moore River, and we tried 2 wetlands down on the south coast. And they're about 350 kilometres difference in latitude. In 2018 and 19, we did a trial again, which was a longer trial, and this time we went we actually put animals into the natural habitat at Ellenbrook as a control, and again we tried a north and a south comparison. And then in the most recent one, which only began a couple of months ago, we've done a largest trial yet down in the area of East Augusta. So I'm just going to show you a few sort of highlights from each trial. So the first trial involved 35 juveniles. They all come from the Perth Zoo's captive breeding program. They're around about anywhere between 2 to 8 years old, depending, and we basically release them when they're at a certain size where we know that their survival is quite good on release. And so we had a master's student, Alex Boomer. She was looking after monitoring the sort of releases in the south, and Gerald Cookling was monitoring the release up in the north. And between them, we collected a whole lot of data just on how well they grew in the first spring hydro period, because we wanted to know that they could grow well in these cooler southern locations. And basically the data that they collected showed that at Meerup, which is one of the southern sites in green, they didn't grow very well, but they grew, and they showed similar trends over in East Augusta, but East Augusta was different to Meerup and had a much stronger growth of those individuals towards the end of the hydro period. After here, this is when the water dries up. And so you can see Moore River, this northern site, they also grew as well, but they stop growing when the water runs out. That's when they have to go off and estivate. But you can see in East Augusta, there's a high rainfall area, so the swamps are holding water for a lot longer, and that allowed them essentially to catch up and achieve the same size. So that was a really promising result. But we were really interested in trying to think, why didn't we get the two similar results in the south? Why were they so different? And luckily, in the year of that study, we also had an Austrian master's student, Katja Smoltz, working with DBCA's ecologists, and she was looking at what was actually the food sources for this western swamp turtle in these different release areas. And to cut to the chase, the main difference we could detect was that the East Augusta site in particular had a lot of tadpoles at the end of the hydro period when the waters were warm and when turtles were really active and able to eat. And we think that's how they did a really good sort of growth spurt at the very end of that 2016 trial. But the trial was a trial, and the deal was that those animals were returned to the Perth Zoo, and the recovery team was going to sort of have a think and decide what the next step was. And one thing that we noticed is that in 2016, it was actually a really unseasonably cold spring, which was not a great time to do a trial of assisted colonisation when we were a bit concerned about the thermal suitability of southern climates. So that was one of the reasons why we went back to the Meerup area again for a second trial. So trial 2 involved more individuals, again, all from the captive breeding colony. This time we have a different student, Siobhan Paget, and we also have put some gadgetry on the backs of these turtles, which is this little tag here, which is designed to essentially record at very high resolution the animals' movements and behaviours. Well, actually, it doesn't record those things. It records 3 different signals, but from those signals, we try and interpret what the animals are doing. So it's recording the pressure, which tells you if the animal's in the shallow or the deep water, tells you the temperature, and it tells you whether the animal's come out of the water and is basking potentially, or whether it's still in the water and foraging. And so Siobhan has done a lot of fancy analysis and an interpretation of these codes, because what we were trying to, or these data sources from these tags, because what we were trying to determine was whether or not if you move a turtle into these cooler southern climates, are they actually as active as they can be in the north? Because if they're not as active, it might affect how much food they're able to eat if they're actually too cold for foraging. And so what she determined was that they do show a bit more basking behaviour in the south, potentially to warm up. And she also showed that they do have slightly less ability to be active if you're in these colder climates in Mirror. So that sort of supported what we expected. What we don't yet know, though, is how limiting that activity is and whether it's actually going to prevent them from accumulating enough food for growth and reproduction. But overall, the second trial compared to the first trial, because we had 2 sites in common, we got very similar results. We could show that they grew okay in the Moore River, didn't grow well in Mirror. So that meant that the recovery team re-evaluated Mirror as a potential site for release of this species and decided it was not going to be suitable. There are a few other reasons why it was going to be difficult too, just like difficulty of controlling fire was another factor that we considered. So we went back to East Augusta, which was, I guess you remember, was the area that was really promising from 2016. This time we have Bethany Nordstrom, who's a PhD student in our lab. She's going to be working on this population or this particular area in really great detail to try and understand if this is a suitable site for the western swamp turtle. So in 2020, Beth looked at different wetlands in the Scott National Park area. She was collecting data on sort of water temperatures and pH and salinity, and then a whole lot of information about what food sources were in the swamps, and also what other threatened species might be in some of these systems, because there are some threatened fish, and we wanted to know where they were and whether or not, I guess, that might come into our consideration of which site to choose. But in essence, we were looking for some swamps that had the warmer water relative to other swamps, and they had plenty of tadpole biomass. And so what we did, this is just an example of one of the sites that Beth looked at and presented data to the recovery team. This shows you the site that we looked at in 2016 as a really successful trial site. It's just over here in this drone footage. So we went back to this site in 2021, but the problem with this site long term is it's quite close to roads, it's quite close to private land, and it's not a very large site. So we don't think it's a good site for an introduction of this species. But we have found a site nearby in Scott National Park that's looking really good, further away from roads, and has a bit more area for the swamp turtles to establish. But in this current trial, what we've done is we put them back into this test swamp from 2016 because we know this area works, and we've tried this new site in Scott National Park. So trial 3, we've involved, hopefully you can hear this soundtrack here. We had a really fantastic participation from the traditional owners. We had an actual meeting of 2 traditional owner groups. We had the Wajak people represented by Neville Collard here, who's doing the smoking ceremony. And then we also had representatives from the Southwest Bujara Noongar group, the Webb family. They were there as well to welcome the turtles to country. So it was really great to have their involvement and their blessing of this introduction into their country. So this is just showing you some pictures from the release inside Scott National Park. And again, we've put some different tags on the turtles because we're still trying to understand their behaviour in great detail to see just how much activity they can sustain in these cooler environments and whether or not we've got evidence of them feeding as well. So now we've got different types of tags that were attached to the turtles that are actually transmitting their data live to this receiver towers, which we've set up in 2 wetlands. And Bethany's PhD is going to be analysing those data sources and also looking at the diet of the western swamp turtle in these areas, just to try and really understand what their potential for growth is in the south. Here's just an image of, I guess, showing a turtle being monitored in the wetland 1. And again, just showing you on the back there what the tags look like. So we're hoping that those tags will stay on the turtles for most of this hydro period to get some really detailed data for quite a large release group. So what we're trying to do is to put all these data sets together to try and predict what the energy budgets of turtles will be across different climatic areas. So we've used something called dynamic energy budget modelling, which we can actually simulate an animal's energy budget right throughout its life. And what we can do just simplistically is to make some assumptions about how much feeding time they have in different locations, what their temperatures are when they're estivating and when they're in the water. And from those pieces of information, we can basically make predictions about the growth of different females if they were released into different areas. So this gives us a way, I guess, of looking into the future and getting some confidence that we might be able to confirm that reproduction is possible in these cooler southern climates. And basically what we can show of these southern areas here, there will be some differences in how they grow, but overall, because they have more time for feeding, we think they're actually going to do quite well in these areas. So the focus of what we're doing now is we're really trying to understand, I guess, can we be quite confident there's going to be enough energy and enough activity potential for versus swamp tortoises in these cooler environments. These little donuts on the side here just show the differences in climate. They are quite different climatically, just a lot more hot days in the north than you get in the south, and a lot more rain in the south. So it isn't really an analogous climate right now. We are putting the turtles into something quite different for them. But as I mentioned before, we expect that in the future decades, the climates in the south will be much more like the climates that the species has evolved under. We really want to be able to show that females can reproduce, predict how old they'll be when they can first reproduce and what their fecundity will be. And once we can do those sorts of predictions, we can then build population models to just be quite, I guess, clear that the species has a future in a slightly different climate zone. And of course, a major question is, if we go ahead with an introduction and the turtles establish well, are they going to fundamentally change these wetland ecosystems? And that's going to be a very important question to understand. We've done some models again about understanding their impact. We're pretty confident the impact will be fairly minimal. We already know that there's long-necked turtles in these swamps, and we know that the western swamp turtle diet is very much constrained to insects and tadpoles, which are abundant in these areas. So we don't expect to see major ecosystem changes, and that's one of the reasons why this has been such a, I guess, a great opportunity to try assisted colonisation. And I really want to point out that in Australia and New Zealand, we've actually been doing assisted colonisations for a long time. So despite the sort of global, I guess, concern that this is a radical thing to be doing as a response to climate change, we're quite comfortable putting species into new habitats to mitigate the threat of invasive predators. Australia and New Zealand have done this many, many times for their threatened fauna. Australia's done it particularly for mammals put onto islands. New Zealand for things like their birds and their threatened reptiles have also been put onto offshore islands. So in our countries, at least, we're quite comfortable with the conservation introductions outside of range, but we haven't really seen it done so much in Australia or globally, where translocations are specifically addressing the threat of climate change. And that's why this western swamp turtle story is really such a great case study for, I guess, all of us to learn from. And one of the reasons why they're probably not going ahead, you don't see a lot of assisted colonisations happening right now, is I guess the risk assessment is more complex because you're trying to understand or anticipate how the species will perform in a novel environment. You often, as our study has shown, you really don't, there are a lot of decision tools out there that help people make decisions about assisted colonisation, but often they'll need to be fit to purpose and customised. In our case, we needed to have a really good way of predicting future introduction sites using a quite different modelling framework that took a lot of time to test and refine. And as I referred earlier when I was talking about the frogs, there are going to be, in many instances, legislative, political, ethical and philosophical barriers to how people perceive assisted colonisation and novel ecosystems that might prevent some of these projects going ahead immediately. But I just want to point out, just to finish, what the characteristics of species that I think are going to mean they're going to be good candidates for considering assisted colonisation in Australia, and the western swamp turtle meets most of these. It's when climate change is, I guess, perceived as the major threatening process is where this sort of thing will get great support. You need to basically demonstrate that the species does have emerging habitats suitable elsewhere. Obviously, like the moss frog, there was nowhere to go. But if you do have a species that has areas that should be suitable for it, then that's a species that you could consider for this type of conservation introduction. You also might want to be sure that the species won't evolve on its own and be quite actually have the adaptive capacity to adjust to a new climate. Where the species with a long lifespan, like the western swamp turtle, we don't think it's going to have any chance to respond to climate change in an evolutionary sense, which is why it needs this sort of assistance of relocation. And of course, the swamp turtle has significant barriers to dispersal due to lack of habitat connectivity, and that's probably going to be the same for a lot of threatened species where this might be considered. The other great thing is having a captive source population made it a lot easier to try this sort of approach, because we weren't talking about moving animals from their core natural range. We had animals specifically bred for translocation projects. And we knew that translocations were successful because DBCA have been doing this for about 25 years. And the other reason why this was a really good and suitable species to do it is because we had such strong scientific collaboration, strong support from DBCA, and strong support from traditional owners as well to go ahead with these sorts of projects, which I think are going to be critical. All right, well, I think I'll wrap up there. And this final slide is just acknowledging a lot of the collaborators across the 5 projects that we've talked about today. And I really just want a big shout out to the Department of Biodiversity, Conservation and Attractions, because we've really collaborated closely with a lot of the researchers there for at least 3 of these projects.